So thank you for the introduction. So I'm Jean-Charles Gautier. I will present on behalf of the committee. We'll start with a review of the current charter period and proposed plan for future work during the next charter period. So slide number 2. Thank you. So here it is the membership of the HESI Committee on Biomarkers of Nephrotoxicity. There are several pharmaceutical companies which are listed on the left side. There are also several collaborators. Another consortium, it's a PSTC Nephrotox Working Group, and we're also collaborating with the National Center for Safety Evaluation of Drugs in China. And the leadership team is Ernie Harper and myself. And there was also Ted Locke up to beginning of the year, and Regan from HESI is managing this committee. Next slide, number 3. So slide number 3, it's just a recap of the mission of the committee. It is first to advance the scientific basis for the development of application and of biomarkers of nephrotoxicity. The second is to develop a systematic approach based on newly available technologies for identification and evaluation of biomarkers which would bridge between preclinical and clinical stages during drug development. And the third objective is to provide a scientific forum for building consensus on how to apply biomarker of nephrotoxicity for risk assessment. Next slide, number 4. So you can skip this. Number 5. So just to introduce, now we will review the current status of the current charter period, which was from 2012 to now. We had 3 main activities, and the main focus was on the evaluation of miCRNAs as kidney biomarkers. We had, besides this main topic, we were involved in technical best practices in relation with biomarker evaluation and interpretation. And the third part was to complete a work which was started some years ago about the gender effect and the reversibility study, and it is about completion of manuscripts. Slide number 6. Next slide. So going directly to the goal, which was the third one on the previous slide, it is on the follow-up on previous qualification efforts regarding kidney safety biomarkers, on evaluation of gender effects and prodromal reversibility in rats. And just for the context, it was additional gentamicin rat studies which were designed and conducted several years ago in order to address questions from health authorities, FDA, and EMA, which were asked following initial biomarker qualification studies. So the outcome is that we conducted two studies, one which focused on gender specificity study, which was conducted by Sanofi, and the outcome is that we observed variation of baseline levels for some biomarkers in males versus female rats, and we observed very similar patterns of biomarker induction after treatment between males and females. 2 publications were issued on this work recently. It was last year. And the second part was a study which was conducted by Pfizer with gentamicin in order to evaluate the behavior of biomarkers of nephrotoxicity for prodromal and reversibility study. It was a 6-week study. And the main outcome was that we observed patterns of biomarker expression and then recovery which were significantly different across the different biomarkers evaluated. And a manuscript is in advanced draft for this part of the work. So it's in nearly completion now. Another point, the last item, is that the data which were obtained on these two studies, they were shared with the consortium PSTC, the Predictive Safety Testing Consortium, in 2010, and the studies were included in the last non-clinical briefing book, which was submitted by PSTC Nephrotox Working Group to FDA and EMA in November 2013, and the outcome of this submission was a letter of support for 2 urinary kidney biomarkers, which were osteopontin and NGAL in the rats. Next slide, please. So here we are on slide number 7. It is about the second part of the work from the current charter. It's about the technical best practices for biomarkers evaluation and interpretation. So we formed a sub-team to focus on these aspects, and this sub-team was led by Yi Yang from AbbVie. And the approach was to have a survey across the committee in order to evaluate to better understand the similarities and differences in the methods of urine collection and creatinine assessment practices as regards to the urinary biomarker measurements. And we identified several knowledge gaps during the survey, and we agreed on follow-up efforts, experiments to further explore first, the effects of food and feces contamination. Second, the effect of urine collection temperature, room versus ice. And the third was on evaluation biomarker normalization, urinary creatinine versus the urinary volume. Next slide. Number 8. And here it is the status for this subgroup. We have completed the analysis of the survey results. We have identified gaps in the course of the survey, which facilitated the design of experiments in order to investigate the effect of temperature, food, and feces contamination on biomarker evaluation. And for this part, the experimental work is now completed. Next slide, number 9. And here it is a high-level summary of the key finding, best practice recommendation on this, which was a follow-up of this survey. So the recommendation is to collect the urine in a metabolism cages over a period of 16 to 20 hours. It's here for the rats. Urine are collected with control of the temperature below 4 degrees, and it is to avoid, because there we observe degradation of kidney biomarkers like clustering osteopontin when you store the urine at room temperature. The third main recommendation is to fast. The fasting is optional during the collection period, but you need to be aware that urinary total protein and glucose can be affected in the presence of food. And the last main recommendation is to avoid, to minimize food fecal contamination because the feces clearly affects the level of biomarkers like urinary osteopontin and clustering. So samples which are visibly contaminated with feces should be discarded. Next slide. So number 10 here. So now we are at the stage of follow-up survey for the normalization practices. It's about in completion. and we still need to conduct some additional work in order to address the biomarkers stability during a longer-term period, during several months, several years, at -80, in order to evaluate the stability of these biomarkers. Preliminary results were presented this year at SOT meeting with a poster, and a manuscript is under preparation. And the anticipated outcome is a better understanding of best practices for sample collection and data interpretation with the end product, a peer-reviewed publication. Next slide. Number 11. So now we are on the main topic, which was worked by the committee during the current charter period. It was on the evaluation of microRNAs as kidney safety biomarkers. And here the goal is to explore the expression of microRNAs as biomarker of kidney toxicity in order to identify tissue-specific or pathology-specific microRNA patterns for drug-induced kidney injury in urine samples. The rationale is that microRNAs could be a potential alternative approach to protein biomarkers for several reasons. First, they have a very good stability in the urine. We can also develop assay with RT-PCR, for example, which are relatively easy and fast to develop. There is a good similarities of microRNAs between species, so potential for translatability and up to human. And one also very important point is that several publications have showed that microRNAs expression is distinct among different anatomical regions of the kidneys. So all this reason can justify that it's important to work on this topic. And also the last one is that there is a possibility to address as yet unmet biomarker need, especially for glomerular injury, for which we have not yet good biomarkers to monitor this type of injury. And also there is still a need for biomarker for distal nephron segment. Next slide. So here is a slide which, in order to provide some high-level background information on the role of microRNAs in kidney physiology and pathology. So it's just another justification to work on this type of biomarkers to identify a biomarker of kidney injury. MicroRNAs are regulator of key renal function, and this was demonstrated by depletion of microRNA experiments in rodents with dicer knockout mice experiments, which indicated roles for microRNAs, especially during kidney development, maintenance of renal function, and pathogenesis of renal diseases. Just for example, in diabetic nephropathy, miR-192 is a critical regulator of co-collagen production in this disease. It's just an example. And there is also evidence of differential expression of microRNAs in several renal diseases, for example, glomerulonephritis like lupus nephritis, IgA nephropathy. Next slide. So it's important to work in a context of a consortium in order to share experience on this topic, because there is a limited experience to date with urinary microRNAs as kidney safety biomarkers in most organizations. And so it is important to share experience, knowledge about best practices for experimental design, execution, and data analysis. And another benefit for working in a consortium approach is to interface with another committee, the HESI Genomics Committee, in which a program is assessing pre-analytical variable for assessment of microRNA in biofluids. So we can benefit from each other about this type of experience. Next slide, number 14. So our approach was to evaluate, to test several nephrotoxicants affecting the three nephron segments, which are glomerulus, proximal tubers, and the distal nephron segment. Next slide. And here the goal is to identify site-specific microRNAs for different segments. And the approach was to run several in vivo studies in rats with both reference and proprietary nephrotoxicants affecting different segments of the nephron. We evaluated the pathology with the kidney sampling, microdissection, or macro dissection. We conducted kidney histopathology. MicroRNA were profiled, and selected microRNAs were measured urine quantitative RT-PCR on tissue samples. And we collected the urine, measured standard analysis parameters, urinary safety protein biomarkers, and of course we measured profiled microRNAs and selected microRNAs by RT-PCR samples. And for this, we usually use several methods, the TLDA cards and other techniques. Next slide. So number 16. This is a list of the study which were conducted across sites on microRNA for a microRNA evaluation in rats. There are several nephrotoxicants affecting different kidney segments, glomeruli, proximal tubers, distal nephron segment, and the workload was shared between several companies. Most of these studies have been completed now, and the microRNA have been measured and are under analysis. So here it is just a slide number 17. It's just to provide an illustration of the type of results that we obtained. It's about a profile of potential site-specific urinary microRNAs in rats. We compared a study conducted with the cisplatin, which is a proximal tubular toxicant, and versus a compound with a pathology, which is a nephrotoxic serum with antibody against glomerular basal membrane, which induce glomerulonephropathy. And you can see on the left, it is a Venn diagram. We had 68 microRNA which were changed in the urine, specifically in the cisplatin study, and we had 6 microRNA which were specifically modulated in the urine in the NTS study. And on the right, you can see the hierarchical, the heat map for this modulation for cisplatin with the 3 milligram per kg per day dose. And on the right, on the far right, some miRs, the 6 miRs, which were modulated specifically in the glomerular injury model. So these miRs could be potential candidates for site-specific modulation. So the status, we are on slide number 18 now. The status is that all in-life studies have been completed. Both microRNAs and corresponding clean path protein biomarkers and histopath evaluation have been completed. And the committee is currently working to collect and compile the study data across site for meta-analysis. And for this purpose, we had collected histopath data using an harmonized lexicon from PSTC in order to compare across studies. There are several approaches to microRNA normalization which have been reviewed, and further work is underway to determine the type of analysis which will be applied for the meta-analysis. Promising microRNAs have been identified from the studies, and for example, cisplatin studies suggest concordance of several miRs which were identified by at least 2 sites. 19 modulated miRs were found in both studies, even with studies which were conducted with different rat strain and different doses and protocol. And 5 manuscripts have been published recently on this work, on the result from individual studies, and another one is currently in press. Next slide, number 19. So moving now towards to translational biomarkers, the committee has established a collaboration with the PSTC, and the goal here will be to share. The PSTC is currently conducting studies with large animals, with non-human primates, dogs, and we will share samples from these studies to investigate microRNAs with the HESI committee. And data will be shared between the 2 groups in order to facilitate identification of promising biomarker candidates. And the committee has also been identified as a partner in a project proposal submitted to the Massachusetts Life Sciences Center Cooperative Research Matching Grant Program. Here it is for clinical studies for translation in human. And the plan is to collaborate with the Harvard Medical School and other partners like Biogen and the consortium of FNIH Safety in order to identify and validate promising microRNA biomarker candidates for clinical testing and validation. Next slide, number 20. So here it is a high-level summary of the accomplishment to date regarding the microRNA program. So we had a very strong engagement of 7 organizations across Asia, Europe, and North America to conduct several studies, with an even broader engagement in study design and interpretation. We have pulled expertise towards study design. We have learned a lot about cross studies with regard to technical challenges and best practices. And we have obtained preliminary identification of candidate toxicity biomarkers, microRNAs, in several individual studies. We had a very active crosstalk with the HESI Genomics Committee to mutually inform program progression. We have worked on the strategies for cross-laboratory comparison, normalization in development based on joint experience, and we have established collaboration to extend the rodent microRNA work on to large animals and humans. Next slide. Number 21. So the future work for the committee now will be to analyze all the data across the rat studies and identify potential biomarkers for a site-specific injury. We wish to publish in the peer-reviewed literature the results obtained in all the other individual studies which have not been published yet. We would like to conduct large animal microRNA assessments in collaboration with the PSTC, and we would like to collaborate with Harvard Medical School for clinical translation of microRNA biomarkers. Next slide. Number 22. So it's the timelines for the most recent charter period. I will not go into the details, but just to show that it's on the upper part of the slides. It's about the timelines for the best practices subgroup work. On the upper part, it's about the conduct of the rat studies and the development of a data analysis strategy and collection of cross-study data. And on the lower part at the bottom of the slides, it's the timelines for the work on to develop a collaborative agreement with the PSTC on large animal miRNA evaluation and to further define the scope of work with large animals. And the outcome also was that we have presented this work during several meetings, the SOT, Eurotox, and it was shown on the bottom of the slide. Next slide. So now looking beyond now for the next charter period. Next slide. The recommendation of the committee would be to request a recharter through year-end 2017 in order to complete ongoing and planned work. Next slide. So it's number 26. It's a high-level timelines for the proposed charter between now and the end of 2017 for the recharter of the committee. So going from the top, the upper, and to the bottom of the slides. So first, we would like to complete the meta-analysis, publish findings of rat microRNA studies. Then we would like also to complete the evaluation of renal biomarker assessment best practices and publish manuscripts, and it would go to end of this year to end of next year. And a major part of the work would be to conduct microRNA assessment in larger animal samples. It's from next year to middle of 2016, and then to analyze and publish the work to end of 2017. And also for the human translation part, on the bottom of the slide, it's to mine rodent data for candidate microRNA biomarkers for clinical evaluation and for collaboration with clinical studies. So we envisage to complete this work, all this work, by end of 2017 to have a completion of and the scope of the work and sunset. Next slide. So here there are two last slides just to provide some insight about here on the ongoing and planned studies in large animals, just to show a high-level plan of the studies which are ongoing or planned in large animals, in non-human primates and canines, either in collaboration with NCSCD for glomerular injury toxicant, for doxorubicin, anti-GBM based glomerulonephritis model, and there is also another study which was conducted by Pfizer with the bovine gamma globulin model in NHP. Concerning proximal tubers in non-human primates, there are a study on cisplatin which was conducted by Janssen, a range finding study, and there is a plan to conduct another study, definitive study, by end of the year, beginning of next year. There is currently a gentamicin study which is ongoing for a definitive study with gentamicin in non-human primates, and there is plan to conduct a tubromycin study in non-human primates. Also, all this in collaboration with the PSTC. And concerning the canine, the plan is to conduct studies with cisplatin and tubromycin in the context of the PSTC, and here it is to support the clinical qualification of tubular injury biomarkers. And these studies are under preparation. Next one. So just here to show an example of results, microRNA changes which were observed with non-human primates, Cynomolgus, which were treated with gentamicin with a low dose of 10 milligrams per kg per day, 10 days daily. And here it is the results on day 11, just to show that we observed clear increases of some microRNAs on day 11 as compared to the pre-test here, just to show that it's a proof of concept study, just to show that it's possible to conduct experiments with monkeys. Here it was done with a TLDA from human, which showed that we can measure microRNAs which were valid in Cynomolgus here. Next slide. Next slide. So here it's number 29. It's the last slide, just to show the strategy to evaluate and validate microRNAs as biomarker to detect kidney injury in human. There is a first part on the discovery and confirmation, which is done by the team of Vishal Vaidya in the Harvard Medical School, and in collaboration with Biogen, is to first identify some microRNA candidates from patients treated with cisplatin, and the HESI committee would be involved to propose candidates from the rat studies which were conducted previously. And the next phase will be to validate this marker in human in clinical studies which were conducted in the context of other consortium with FNIH and safety. So in order to validate this, and here we are seeking support, and this is the reason why a grant proposal is ongoing. So that's the last slide about here, and I'm happy to take questions with Ernie on this.